Thanks for joining the session. I think it's the kickoff for the metascience kind of series. I'm Sime Chow, and I'm a scientist. I run a company called Arcadia, where we do research on evolutionary biology. I'm also a philanthropist, so I run a couple of different nonprofits, including Astera Institute. And what I want to tell you about today is something that started because basically I had a baby. And there is a point to this, which is not about the baby, but that during my maternity leave, I had a ton of time to kind of like take a break from my day-to-day of running stuff and think about, as a philanthropist and a funder and as a scientist, how I might blend these to think in a more opinionated way about the kind of science that we build. And this is kind of a larger question that I'd like to come back to at the end of like how this happens more. Because I think sometimes too often there's a separation between sort of funders and the people doing the technical work, and that leads to very different outcomes. So really what I will spend most of my time talking about is PDB 2.0, which is kind of what I landed on because I was trained as an X-ray crystallographer. Hi. And so I kind of went back to my roots during my maternity leave and thought about like, what would I be pitching myself right now if I were on that side of the table? And this is the result of that. So PDB 2.0 is thinking about the kinds of data that we actually need, not just a scaling problem, but the right data that you might need to break through with the next model for protein structural dynamics. And that, I think, is really the next frontier. So I think one of the big picture things motivating this, since we are in a more meta session, is just how the future is already here, that we need an entirely different way of doing things than 50 years ago. And there's a lot of like basic differences between now and then that might cause us to rethink every aspect of how we do science. So one is that like we're not working alone as humans anymore. Our frontline operators are agents. So what does it look like as that becomes increasingly the case? How we publish, who are we even talking to, what data is like useful for that? That is an entirely different question than if your frontline operators are only humans. Data is king, right? So like, how do we get more of the right kinds of data out there so that when we think about using AI and other technologies that it can actually be productive? I think ultimately everything needs to move towards being more radically open. If we want to think about building a knowledge corpus that can actually, in the speed that we need today and can actually do science today, is informing a lot of the new models and the intelligence that's going into that. Scale, like no doubt, like with machines involved, we can think vastly differently about the amounts of information we can synthesize and also the speed of that. And all of that is also affecting how many people can actually participate in the scientific process. We don't need sort of these silos and a few individuals in the world acting on things. We can all be contributing to all the different outputs that allow this. So to do that, basically, you have to think from the very root of a system that you're setting up to really reimagine how you engineer all of these assumptions in better. And I think science is going to change dramatically over the next 5 years because of that. And so this actually feels like forever ago, right? This is 2021. It's not forever ago, but this was a huge moment for science, that sort of the first domino to fall. This was Google DeepMind announcing AlphaFold. And there's just a few things of this image that are important to think about. One is that it was announced on Twitter. Two is a company with arguably one of the biggest breakthroughs in biology in the last decade, if not more. And also that you had tons of people commenting and interacting with this. And now, we don't even remember where this was published, right? We just know it exists now and everybody's using this tool. So it's just like a huge example of how utility really rises to the top above some of that noise. So the question, though, is like that you hear all the time in these like funding conferences, progress conferences is, what's the next PDB? Really what that question is asking is like, how do we set ourselves up for the next big breakthrough? And how do we think about the right data, how to scale it, how to get participants involved so that it happens much faster than the first PDB? And so I actually have an answer to this that I want to talk about today, which is I think we're not done with the PDB. The next PDB is also PDB, which is PDB 2.0. And that is to now think about how we move from static shapes of proteins to understanding how they move and being able to predict that at a large scale. And so I will just make a point here that I'll come back to at the end, which is like, rather than sitting back and saying, oh, let me just get a bunch of pitches for this, I've started thinking about with our philanthropy, especially because I'm a scientist, how we actually get in there and just sort of like relentlessly go after this problem and get our hands dirty on helping to build this. So hiring out the right infrastructure teams, hiring the right personnel to do it that maybe is like difficult to do in academia. So software engineers, ML engineers, and just really being like opinionated about this and not letting it go until we get it done. So it's a lot more operational than I think is typical of most philanthropies. And so we have Astera Foundation, which is really the main house for a lot of this work. The Navigation Fund is a 501c3 we have also. I mostly mention it because it's supported a lot of open science efforts that are more of the metascience aspect to this. And also our entire endowment there nearly is invested in a compute cluster called Voltage Park that has about 24,000 H100s. And part of that is to provide a critical resource that is important for all work in the future, especially science, where it might be difficult to get access to some of those types of resources. So back to this question, I'm just going to start here. Like, what's the goal of structural biology to really think big about what it is? And I pulled this from one of my past papers. Like, it's definitely not this. Like, we don't actually care that there's like this like loop 1, loop 2. Like, no one gives a fuck about this, right? What we actually give a fuck about is like what that might tell us about what that protein does and what that might tell us about what else it looks like. That again tells about what that protein actually does. So that's the real question, is like, what are these things doing? And so how do you actually get at that? And I think like it's helpful to think about this by thinking about protein by numbers of like what other information is missing. Proteins have very busy lives. They take up over 50% of cell mass by dry weight. There's like billions of molecules per cell, as well as millions of distinct proteins acting in a teeny tiny cell. So there's a ton of collisions, right? Like Brownian motion is causing a ton of collisions and ton of motion that they can actually like move microns within seconds across a cell as well. So this is all to say that what we're missing is a lot of context that matters. The first piece of context that's missing is actually just how these shapes move. And so like a great illustration that I just want you to take home with you is like everybody, I think almost everybody, and we're a pretty nerdy crew, so maybe not, but knows what this silhouette is. Does anyone not know what that silhouette is actually? I think maybe you won't admit it. But so we all know this is Michael Jordan. This is like classic dunk, right? And the reason you know that when you see that silhouette is you're seeing like the motion that's happening here. You see a static snapshot that's one in a series of things that actually tells us what he's doing. And so the question we're asking today with the PDB is like, how do we move from these predictions about these pictures to living movies of what they do to start getting even closer to the functions of proteins? And so a big complaint you might hear from people is like, X-ray crystallography is not going to get us there because it's giving us static snapshots of a protein in a crystallized form. Well, I would actually pause here and try and question that. I would say we derive static snapshots from X-ray crystallography data, but there's actually way more in that data that we toss out because this is what we do. And so I'd like to talk through some of that to the best degree that I can in the time allotted, like why I think that's true and why I think that means we got to go all the way to the root of like, what are we even gleaning from the data to get the right stuff? And I think there's a version of this for every structural biology modality there is, NMR, EM, like we're not quite collecting the right information. And so more of the wrong information is not going to get us anywhere interesting. And so here's actually what, if we can work backwards from an AlphaFold prediction, here's what you're actually getting. It's not ground truth, actually. It's pretty close to the crystal structures you can experimentally generate. That is definitely true. There's very little error between those two things. But then you start going back and you say the models we get from crystallography experimentally, there's like up to about 20% error between that and the electron density map that's generated from diffraction data. And then if you go backwards from that, there's more and more compounding error as you go back to the original crystal structure. So actually, like, we're not getting ground truth from crystallography data at all. And there's a lot of really interesting pieces in there that are relevant to the question of dynamics. Most of all, I would say, is the fact that inside a crystal, yes, you have mostly one conformation. That's how the crystal can form. But there's actually heterogeneity in that crystal. There's a lot of other conformations it can take. And we just like process all that noise out and try and solve for the consensus structure that's in that crystal, which is why you have this much error coming out, because it doesn't actually match the ground truth of the empirical data that you get from the outset. So what's the solution to that? It has exactly to do with what we're like optimizing to measure in this. So what you see here is a classic X-ray diffraction pattern. The spots on it, which you think are the signal, are Bragg scattering. So those are the like strongest peaks from your consensus structure. That's enough that it contributes to such a strong signal. And so everything we do in crystallography is sort of optimizing for this. You're trying to find the right proteins, the right mutant, or maybe the right crystallization conditions to maximize those spots. So you're selecting for conditions that give you a consensus. The problem with that is like in the background, you can't really see it in this image, but if I invert it, there's this halo effect. There's like a whole pattern in there, right? That is the heterogeneity, which is all the other conformations that exist. And there's enough experimental evidence now for us to know that those conformations are actually relevant, that you can patch those together to create more of a statistical framework about the range of conformations a protein can take that often very well match the real motion that they do in vivo. And so that is a totally different question now, really. Like we don't, let's say we don't care about Bragg scattering. Let's say we care about the background, which is called diffuse scattering. We're just like leaving all that data on the table every time we collect a data set for crystallography. We just throw that away. So what do you have to do to like pull out what's hidden data, which I actually think stands a much better chance of getting us to protein dynamics modeling than those peaks that are the consensus. And so it's actually not a very easy problem. And, you know, when I first started during my maternity leave, basically I would just like cold call people, anyone that worked on anything around this, I would call them like, what do you think we should do? And I wasn't getting any like good answers. It was mostly crystallize more proteins, do more things like that. Kind of makes sense given, you know, their standpoint, you know, but what they don't feel like they have the ability to break through of is like there is a whole pipeline of dependencies for this particular way of doing things. It's not like if you change the beamline hardware for doing this, it changes any of these other things. So you'll immediately run into your very next wall. First of all, I think it helps for me to be like, look, I'm going to fund this only if we do something interesting. What are your real bottlenecks? And so I was like, and who else should I be talking to with all these different steps? And so basically what we did was we thought about the holistic redesign that would require not only grants to a bunch of these people, but a coordinated effort where they're iterating together in a whole feedback circuit at once. And so rather than doing like 500K, we ended up doing 5 million across a couple of different groups that represented specializations across all of these. And now we have a whole shared Slack channel. We have like weekly goals, and it has to work across the whole loop of it to collect data differently and rethink everything from even the samples you prepare all the way to like how you validate any of this through modeling. And basically like you have to do it all at once. And that's led to entirely different conversations about what each of them should do, because they're no longer thinking within some random constraint of like what they're going to run into next, right? And so that's been really, really important. And so we're also doing it across 3 different US beamlines, because normally, I don't know if y'all have ever been to a beamline, but it looks like it's like patched together with duct tape. And like normally you can do some random thing on one beamline and there's no way to get it to work on another one. So already it's just like not scalable. The other thing we've thought about is, you know, what's at stake here is basically if this goes well, and we have some early signs, we've only been operating for about 4 months and we're already putting out publications and data sets and other things. But if it actually works, you don't just want to then fund this set of people to generate all the data that's necessary. It's like not actually possible to do that, right? To break through to a model. What you actually need to do is figure out how to make it so easy and useful to everyone doing structural biology, even a random biochemist who doesn't normally do structural biology. When they go to the beamline, they should be doing it this way and getting better results. And that's how you scale it. And that's how the PDB actually scaled it, right? Because it's like the whole scientific community started operating in some new way. And so basically we like earmarked enough funding that if this goes well, we could potentially fund the upgrade of every single beamline in the US to now operate in this different way. And a cool thing that we're finding now is that like, and this kind of makes rational sense, right? That by virtue of angling for the hardest data in there, which looks like noise, even the Bragg solutions are performing better statistically in terms of actually representing the data well. So I think that makes it very compelling for everybody then to start adopting this if we can make it really easy. I think this is a big thing we're going to have to think about in science now, because if we're thinking about redesigning systems, you have to tackle multiple dependencies at once and they have to work with each other because they're part of a whole. So this was launched, like I said, in June. One of the things that was really different about this is that we really co-developed this, and it took a lot of trust, Like, they basically got a random cold call in their office from some lady that was on maternity leave. And so, like, I had to, like, have a lot of meetings with people, bring them together, and really also get them to trust me. And I think it helps because I'm a crystallographer, like, so we could, like, speak the same language on this. And I don't know how to scale this. Like, Pranay and I were talking about this. Like, this is an extremely manual process. I don't know what this looks like at a federal level or whatever. And I don't even know what this looks like maybe at the future of a Sierra, but, like, it's a thing we're going to have to experiment with, is how you actually get really serious about these technical problems and have active scientists also thinking about the funding piece so that they're really empowered to think differently. And so we are starting with X-ray crystallography, but we're going to start moving out to other modalities. The team has about, like, I guess 16 months left now to figure out a first series of couple questions that will help us determine whether we want to move forward or not. They have a ton of real-time output, so they're, like, blogging about their stuff already on their website. They're already putting out data sets. We have a whole logbook. They just put out the newest post was actually just an automated conversion of Slack conversations into, like, how they're thinking about it. It was actually really cool because it was like, this guy thought this guy disagreed. Like, here's what they're going to do to test it. Like, that's really valuable stuff in terms of the real intelligence data that we need to ever develop autonomous models that have scientific intelligence. We do a lot of centralized support for them. So we are hiring, like, software engineers, other engineers, compute, all kinds of other infrastructure, including, like, publishing support for doing things a little bit differently. None of the things they're doing are going to appear in journals anywhere. And that was a very important forcing function to get them to think totally differently. The cool part is it got them to think totally different, even about their scientific plans, because they're just like, screw it. Like, why? I'm not going to do this. There's no point if there's no journal article. Let me just actually do the things that I need to do to get this done. And I'm putting a lot of pressure on them on the clock, not a lot of pressure on them on the money. Because it's like, I don't want this to fail because you took some shortcut and we're cheap about this one thing, right? Like, this is, like, kind of like understandable in the venture space, but it's a thing that you kind of have to train academics on. So what's cool also is, like, we have representation across a couple of beam lines, as well as, students and postdocs in these labs, all of which have agreed enthusiastically to not publish their stuff in journals. And I think that's really cool because 5 years ago, I would have never been able to convince a single goddamn academic to do that. And now they were not only willing to do that, they're excited about it. And I think their science is better for it. And ultimately, also what that means is there's no more leaning on those kinds of impact factor proxies. Like, they got to make this useful. They got to make it so useful that it rises above all of that. And that's a really fun goal for the right people. So, like I said, they have a seed for about 18 months. It is a hybrid model of a distributed team across different institutes. We can provide a bunch of stuff. And mainly, honestly, the thing that I provide, which is, I think, also maybe the most possible failure mode, is coordination. It's a complex project management task in front of us. Just to tease you about, like, different ways to think about publishing, right? Like, one thing that we're doing a lot of at Arcadia, as well as in this project, is, computational work. So there's Jupyter notebooks for a lot of that work. Normally what you do is you do a bunch of this shit, and then you do a bunch of manual steps, which is, like, not even reproducible, and it takes forever. And, like, why is it a different step than the other thing? So how do we collapse those into a single thing? We started writing code to convert them automatically into the manuscript. And the conclusions update, the graphs update. And actually what's also really cool is, like, people can take your Jupyter notebook and plug their own data in and ask for yourself, like, do you agree or not? And it's actually falsifiable and reproducible in a way that would not be possible if we're just writing basically a whole PR segment after it. That's, like, not the same thing at all. And a lot more work is in silico so we can do this. So I think it would be really fun to get more people to try this. It started out as a thing that someone in my organization tried just on a whim. And then, like, everybody in my organization only wants to do notebook pubs now because it just is easier and it's faster. And I'm really proud of that. And so Diffuse is trying new things like this too. And so I guess this is also just a call for, like, if y'all want to try anything different or funding people trying something different, we're so game to experiment on this because I think, like, we right now, especially with all the changes going on, it's looking more and more like journals are no longer going to be the main, like, medium by which science happens. what are we going to replace it with is actually the question we should all be grappling with and trying to move quickly on, because we could definitely end up in a worse place than we are today if people don't get with that and start accepting what's happening and trying to experiment with something else. So I guess I would just end with saying super important to us, and I think this should be important in metascience, is like we have to think about how the science has long-term utility that is very clear. Otherwise, there's no way for us to ever know if any of our metascience efforts are good or bad, right? Like otherwise, like getting better journal articles is not actually going to tell us if your metascience approach is good or bad. It's just telling us that you like journals. And so, yeah, like something has to rise above that noise. And I think, as far as I can tell, the only one that makes sense to me is like utility and figuring out how we get to a real outcome that can be obviously felt by everybody in the work that they do.